Hello, friends. Welcome to yet another sneak peek into our biology and bioengineering summer camp. In this summer camp, we'll learn about the world of biology and bioengineering in the 21st century. Let us meet our guest speaker, Dr. Rahul Purwar, who joins us at the summer camp from Indian Institute of Technology, Bombay. Dr. Rahul Purwar is an associate professor at the Department of Biosciences and Bioengineering at IIT Bombay and founder of Immunotherapy Cell Therapy Private Limited. He holds a PhD degree in molecular medicine from Hanover Medical School, Germany, and has completed his postdoctoral training from Harvard Medical School, USA. He has worked as a scientist at the Department of Translation Medicine, Immunogen Inc. at Waltham, USA. His main area of research include CAR T cell for cancer immunotherapy, as well as T cell mediated inflammation. Dr. Purwar has developed an indigenous platform for CAR T cell therapy cancer treatment and is committed to bring CAR T cell therapy to Indian patients at affordable cost. His research work is published in highly prestigious journals like Nature Medicine and Journal of Immunology. He has been awarded with several awards, one of which is the Melissa K. Bambino Memorial Award by the Skincare Foundation, USA. We are really excited to have with us Dr. Rahul Purwa. See you at the summer camp.
All right, so we are very pleased to see Professor Rahul Purwar here. He is one of the pioneers in the cancer immune therapy and CAR T cell therapy. In fact, he has reached to the clinical trials now, which is a great accomplishment, moving from the basic labs to the clinical translation in Tata Memorial. So I'm sure you're all going to be very much mesmerized with the kind of work which he's doing and going to appreciate that how well one can take the basic biological leads forward to the clinical translation value. So over to you, Rahul, now.
Thank you, Sanjeeva. I'm trying to share my screen.
Yes,
Can
it
can
works.
you see my slides?
Yes, yes.
Okay, perfect. Okay, so first of all, thank you, Sanjeeva, and good morning to all the students and the participants. So I am assuming these participants are from like is college, right, Sanjeeva?
Actually, the variations are quite a bit from ninth class to BSc.
Okay, okay, perfect. Okay, so thanks a lot, Sanjeeva, for this opportunity uh, to share some of our ideas in cancer immunotherapy to this wide audience. So good morning again, everybody. So as Sanjeeva mentioned that I am one of the faculty members and his colleague in IIT Bombay. And I will be talking to you about this cancer immunotherapy, which is one of the revolutionary cancer treatment. Okay. And in this presentation, I will tell you more like few stories instead of giving you more scientific data so that all the participants can get a feeling what is cancer immunotherapy at the end of the talk and why it is called revolutionary cancer treatment. So there would be less science in it instead of more like a concept and more like a idea to give you like what is called cancer immunotherapy. Okay, so many of you might be reading this newspaper or the going online to read the news, and many times this very like very exciting news coming. For example, BBC News very recently had this headline saying immune discovery may treat all cancer. Okay. Similarly, on the same time, another very powerful news channel or news media, this website Newsweek, they said one size fits all. Cancer treatment could be on the horizon after scientists discover this new immune cell by accident. Right. Similarly, you will, if you see on the online platform or in the news, you will find so many of this exciting news saying that yes, immune therapy, this new modality of the cancer treatment that can treat basically all type of cancer. And many of you, if you are reading the newspaper or interested in this little bit of immunology, You may be wondering, what are these modalities? What are these? How do they work? Right? So this is what we are going to listen. So before we go right into the cancer immunotherapy, just to bring all of you on the same platform, just to give you an idea what is immune system and how you think immune system can fight against the cancer, right? One of the thing all of you may realize, even in COVID era, the immune system, a healthy immune system is extremely important. So if you see COVID-19, what you are hearing that immune system is overreacting in many cases. And that's something like cytokine release syndrome. This word is now popular in the right now, CRS, where patients develop very robust immune response, and that is causing lung damage, that is causing a lot of lung inflammation, and people are dying. Right? So immune system is very robust. But one thing now you may be realizing, a good immune system is important, not just simply a very hyperactivated immune response is not a good idea. So that means you should have a healthy immune system. Immune system should be only active against the something which is dangerous and should kill that particular pathogen. That's what immune system is all about. Immune system recognize the self. Self means our own body part and does not react on it. Immune system has the ability to recognize the non-self, that means all other pathogen, cancer, etc., which are not belong to our body, and kill it. That's the good immune system. But, and in fact, that's why 99% of all of us are healthy, right? Because immune system is doing its job, continuously fighting all the environmental pathogen, all the environmental insult, whichever we are going through. Immune system is protecting us. But in some cases, in a very, very few cases, a dysregulated or a immune system is going haywire. It is not working the way it should work. And that's the reason the diseases happen. Okay? So this concept that immune system protect us and fight against the cancer started right way back in 1891, where the surgeon from Memorial Sloan Kettering in New York, William Cooley, he used certain bacteria. Bacteria was aspirogens. He killed those bacteria, and these killed bacteria he used to activate the immune system. When he used these non-self kind of bacteria to activate the immune system was able to kill many different type of cancer. And that gave the hint for the first time that by just simply activating the immune system can cure the cancer. Okay. And that concept led for William Cooley to be the father of immune therapy. And in fact, his discoveries, remarkable recoveries observed in the patient with advanced cancer. So that means those patients who were very late stage cancer, they even got benefited by this very simple immune therapy, which he started way back in 1891. Then the late 19 or mid 1950, this theory come that immune system or immune cells, they patrol throughout the body and they surveil, they do like a police patrolling the city. Our immune cells continuously doing surveillance in the entire body and trying to find the pathogen, trying to find the environmental insult, trying to find the cancer cell and killing it. That's called immune surveillance theory, and that came in mid 1950. Okay. Then came the era 1980 to 2010, when every scientist tried many different ways to activate the immune system to kill the cancer cell. So they generated vaccine. So cancer vaccine came. So many viruses came. So many biologics like proteins, which you may be hearing, they came in the market, but none of them showed very good success until 2011. So this was the very, I would say, sad era, disappointing era of 1980 to 2010 for the immunotherapy, because here whatever the vaccine or other agents were tried in the clinic, they all failed. So people started losing the hope that cancer immunotherapy is a big thing or not, right? And in the similar time, the cancer is very much hyped as a genetic disorder. People said cancer is simply a genetic disorder because there are mutations observed in the cancer, and that's why cancer is simply a genetic disease and nothing related to immunotherapy. Because immune therapy were not working, genetic mutations were found in the cancer, and that's the reason this is the very disappointing era for the cancer immunotherapy until this 2011. This two gentlemen, Alison and Hanyu, they discovered something very unique and very impactful. Okay, and these two guys got, in fact, the Nobel Prize in Physiology or Medicine. They jointly got the, and this is the story we are going to today hear. Okay, what they did, how they did these, and what is the success story about immunotherapy era, right? So they basically discovered, and they got the Nobel Prize for their discovery of cancer therapy, which actually the mechanism of action or the how it works by inhibition of negative immune regulation. And we will learn what is called negative immune regulation. Okay, so this their success story, how the Nobel Prize they won, why they won the Nobel Prize. So you, many of you may be thinking or curious, what kind of research gets the Nobel Prize, right? So that's the story we are going to listen to. Okay. Another revolution happened in the cancer immunotherapy is coming from another two gentlemen. One is Carl June from University of Pennsylvania, and second is Steve Rosenberg from the NIH, National Institute of Health, and National Cancer Institute. These two gentlemen, they discovered some immune cell that can kill the cancer. Okay, and that is called CAR T revolution. So if the time permits and the organizers permit us, we will also hear another story about it. So in general, if you see the cancer treatment, the cancer, the way it is generally treated by radiation therapy. So people undergo radiation, so the radiation can kill the cancer cell directly. Second, the most successful thing is surgery. If you see the cancer, you remove it. But many of the cases you might have heard that cancer is spread throughout the body, and that is called cancer metastasis. And if the cancer is spread throughout the body, you can't use surgery because you can't remove multiple organs. So surgery works very well when there's a localized tumor. Surgery doesn't work where the cancer is spread. Many of you might have heard about chemotherapy for the cancer, right? And chemo is generally given either using tablets. So chemo means chemicals. So these are chemical drugs which are chemical in nature, either in the form of tablets, capsules, or the injection. So these are given. Okay. Another form of immune therapy is they can be either the chemical drugs, they can be the cell, they can be the proteins, they can be the different type of other modalities like vaccine, like killed bacteria, or like nowadays you know the COVID vaccine, they could be mRNA vaccine, they could be the simply killed bacteria or different platform. So immune therapy can be anything, right from the chemical drug to the cell, immune cells, to the biologics, that means proteins or the recombinant proteins or antibody, or can be the vaccine. And all of these immune therapy, they work by targeting the immune system of the patient. What they do, all these different modalities, they target the immune system in a way, they improve the quality of the immune responses, and that immune response kill the cancer cell. So what are the challenges in cancer treatment? So if you see these drugs or radiation, so chemo drugs, chemotherapy, or the radiation, they directly kill the cancer cell. So that's the advantage that you see immediate effect. But many of you might have heard that these kind of modalities, drugs or radiation, chemo drugs or radiation, when they are used, they not only simply kill cancer cell, they also kill the healthy cell. And therefore, patients have lot of side effects. The side effects can be in the form of some of that you might have noticed. People go bald, they lose hair, they have very bad digestive system, because digestive system cells continuously proliferate, and these cancer drugs, they directly go and kill the proliferating cells, or your intestine continuously those cells proliferate and make your digestive system work. And if those cells are also getting killed as a first target, because they are proliferating in nature, cancer cell is proliferating in nature, and these chemotherapy drugs, they directly block the proliferation, means when cell divide from one cell to multiple cells, they directly kill these cells. And that's why there are a lot of side effects of the chemotherapy and the radiation. Okay. But on the other hand, in contrast, if you see cancer immune therapy, they selectively kill the cancer cell, because they are not directly having effect on the cancer cell. They don't kill directly, they indirectly kill. And how indirectly they kill? They unleash the immune system of the patient. So they basically try to activate the immune system, and the immune system kill the cancer cell, not the directly the drug. So an immune system, as you know, is innate or inherent property is to only kill the non-cells. That is the trick here. That immune therapy, they directly activate the patient immune system, and patient immune system then recognize the non-self, recognize the cancer cell, and selectively kill it. It spare the healthy cells. Healthy cells, our own immune system do not kill the our own cell. Okay, so that's the mechanism of action of immune therapies. Immune therapies, again I will repeat, they can be the chemotherapy drugs, or that they could be antibodies, they could be the recombinant proteins, they could be the vaccines, they could be the cell, or the immune engineered cells, or the normal cell. All these different modalities of the immunotherapy, they unleash the patient immune system, and then patient immune system selectively kill the cancer cells and spare the healthy cell. That's why this therapy is having very minimal side effect compared to the chemotherapy or the radiation. Okay. So having this little background, let us hear their success story, which they took from research to clinic. Right? Jim Allison and Tasuku Honyu. Okay. So Jim Allison, actually, he was born in Texas. 2012, he joined as professor at University of Texas, MD Anderson. And then he became, earlier, he basically, the research work, what he did that led to the Nobel Prize was done in Memorial Sloan Kettering Cancer Center in New York, from his stay during his stay at 2004 to 2012. Okay, he did his studies in University of Texas again, and as of now, currently, he is actually professor at MD Anderson in Texas. Okay, Tasuku Honio, on the other hand, an Asian scientist. He is born in Kyoto in Japan, and since 1984, he has been professor of Kyoto University. He was faculty member of Tokyo University, and from 1979 to 84 at Osaka University. Okay. He has been dean. He got his PhD degree from Kyoto, and he had done a brief postdoctoral fellowship or research fellowship in US at Carnegie Institution of Washington, Baltimore, at NIH. Okay. So how the cancer immunotherapy works? As I said, cancer immunotherapy, what everybody else was trying until 2010 was people were trying, as I said, antibodies, recombinant cytokines, different cell type. These all different modalities of the immune system was tried, immunotherapy was tried, and they all, what they were doing, they were trying to activate the immune system, but none of them was giving good job. And these are like peptide vaccine. If you will go in the literature, you will find their peptide vaccine, dendritic cell vaccine, whole cell vaccine, viruses are tried, but everything was leading to poor response or short-term relief. And these two people tried something different. Okay, and what they found out, they identified the proteins that act as a brake in the immune system. So what is the protein that act as a brake? Now think about a car driver, and here the analogy is, think car as an immune response of the cancer. So if you want a very good immune response of the cancer, then you want to activate it, right? The car, if car has to move, think about if you want to drive the car, okay, how you will drive the car. So car, you know, there are two pedals, most of you may be knowing. One pedal where you have accelerator, right? And then another pedal is the brake. If you have a manual car, it may have the clutch also in India, right? But think about this, an automated car for a moment, and it only has two pedals. One pedal has the brake, another is the accelerator. And if you want to let the car drive, what you do? You push the accelerator, so car moves, but at the same time you release the brake, because brake, your foot should be out of the brake, then only car moves. So accelerator, you think as the vaccines, recombinants, all the different type of oncolytic viruses, all these on the left, what you're seeing on the screen, all these are accelerator. They boost the immune system. And think, everybody was just pushing the accelerator, paddling it, and pushing that paddle down, but car was not moving. The reason was, if you are still having the brake in the car, and if the brake is on, car will never move. How, no matter how much vaccine you give, how much recombinant proteins you give, immune system will not get activated, because there are something called brake, and that brake is in the form of a protein in our immune system. And those brake, these two gentlemen identify. So they identified something different, which nobody was thinking. Everybody was thinking, if you want car to drive, you have to push accelerator high. But sometimes one mechanism doesn't work. You have to think something different, right? So they thought maybe there is something called brake is there, and that's the reason the immune system is not working. So what are these brakes? So immune system, in omics class or in omics lecture, you might have heard, or in your class 5th, 6th, 7th, 8th, you might have heard that proteins are the building block unit of life. That means proteins are the one which do all the function. They are functional unit of life, right? So let's hear about what are these proteins called CTLA4 or proteins called PD1, which are acting as a brake of the immune system, which Ellison and Tasuko discovered the immune system brake. So these proteins, they are present on the cancer cell. So when the cancer cells, which is in the here pink, that come in contact with the T cell, if this protein is present on the cancer cell in a very, very high amount, then the T cells, which is the immune cell of the body, which kill the cancer, the function of these T cells goes down. Similarly, if the PD1 is present on the cancer cell, and if the T cells see the PDL1 or PD1 on the cancer cell, they act as a brake. That means T cell will not get activated and will not kill the cancer cell. So that's the mechanism. In our physiological body, these proteins are only present to suppress the immune system once the disease is gone. So generally, whenever any disease come to our body, these antigen presenting cells, they present that insult to the T cell. T cell get activated, and once the disease is gone, when the T cell already killed the disease, let us say bacteria, viruses, anything, cancer, then in these antigen presenting cells, to calm down these activated T cells, they express these kind of break. So in physiological system also these breaks are present, because they are very important thing, because you need to calm down your T cells after the full activation. But cancer used this as a trick to live longer in the body. Cancer says that, okay, I will express more of these break so that T cells cannot even kill me. So that's the strategy, escape. These are called escape mechanism, which cancer uses for its own benefit. Although these breaks are very important for our life, if we do not have these breaks, our immune system will remain overactivated and again kill us, which you heard in the case of, let us say, in the COVID. In COVID case, overactivation of immune system is happening in the lung, and lung getting too much inflammation, and it is getting dangerous to the many people. People are dying. So overactivation of immune system is not good. However, here in the case of cancer cell, this is using that mechanism and as letting immune system suppress and acting this, using these break for its own favor. And what are these break? CTLA4 and PD1, which are the proteins. Okay, so as I said, what these people did, they got some antibodies against these break. If those antibodies for CTLA4, because anti-CTLA4, or for PD1 break, the Tasuku Honio developed anti-PD1 antibody. So they took mouse. So any research when we do in the human, before human, it need to pass to the multiple different type of animals, like in this case, mouse. So mouse were giving this antibody. They were given the tumor. Okay, they were given the tumor and given this antibody. So if you see this graph on the lower right, on the y-axis is a tumor size, like how big tumor is, and on the x-axis is days after the tumor injection. So imagine if they injected the tumor on the day 0, then it took 10 days for tumor to grow some measurable size. Then the group that got this, let us say, another antibody, anti-CD28, it has lot of tumor growing. The control mice that did not receive any antibody, there also tumor is growing a lot. But a group got the anti-CTLA4, which is the break antibody against the break, then tumor was not growing in these mice at all. So this was excellent discovery, showing that if you target these kind of break, if you remove the break, so removal of the break was done by the antibodies against these proteins. Okay, then there was no tumor growing. So that led to the very impactful research. So what resulted into a Nobel Prize? An impactful research, because this research ultimately led to an untold number of lives. They have been saved by the science they have pioneered. And that's what said the Jed Welchuk, who is chief of immunotherapeutics and Memorial Sloan Kettering. The thousands of lives are saved. That's what I want to convey here, that what results in a Nobel Prize. So research should be impactful, that impact the life of thousands of people, and it has to be unique. While entire world was focusing on the boosting the immune response by using vaccine or by recombinants or by using antibody, these guys thought something unique, something different, identified the break of the immune system for the first time. And those breaks now called checkpoints. And if you bring the inhibitor of the checkpoint, so checkpoint here is PD1 or CTLA4, inhibitor drugs, they are called anti-CTLA4 antibody or anti-PD1 antibodies. And these checkpoint inhibitors drugs, they save thousands of lives. And if you see the clinical results of with these immune checkpoint blockers, and they are phenomenal. So they are basically, so now these both the drugs are in the market. One is called, different names are given, and ipilimumab or Opdivo, nivolumab. So nivolumab is anti-PD1 and ipilimumab is the anti-CTLA4. And basically these scientists, they gave the birth to the concept of immune checkpoint inhibition. Okay, if you I don't know, some of you who are in BSc or plus 12, plus 2, um, uh, you may be knowing these graphs. These are called survival graphs, where I am trying to show you these three different groups. Those who don't understand it is okay, not very important. So in this particular slide, we are showing the patients of the skin cancer, melanoma, and they were divided in three different groups. One group was given anti-CTLA4, another was given anti-PD-1 antibody, and there was third group which was giving both the antibody together. And here, patients who survived, almost 60% patients survived, those who received both the drugs. 50% patients survived till 36 months, 3 years after even detecting the last stage cancer, indicating these drugs really do wonders uh, in terms of saving patients' life or extending life by more than 3 years. Okay. So finally, like, what inspired these people to do their research? So what are the things that inspire the Nobel laureates? So in case of Jim Allison, he wrote that his curiosity to learn about the T cell function, curiosity to learn about the immunology. So he said, I'm a basic scientist. I wanted to understand how T cells work, right? And in addition to that, he has a family history of cancer, mother dying of lymphoma when he was 10. And that meant he always had an eye on whether his work might lead to a new therapy. And it was always in the back of his mind. So this kind of curiosity and some kind of motivation basically help him to be focused and do some new research. And his work was published in good journals, very high impact journals as well. Okay. What inspired Tasuku Honio? He began his research after medical school. Classmate died for stomach cancer, and he said he want to continue my research so that this immune therapy will save more cancer patients than ever. And Tasuku Honio works also published in very, very high impact journal, the Immunity, Journal of Experimental Medicine, etc. And in summary, if you see the discovery of immune break, in fact, it saved thousands of lives and thousands of patients with lung cancer, kidney cancer, Hodgkin's lymphoma, melanoma. So all kind of patients' life was saved. And very famous, like US President Jimmy Carter, he was diagnosed in 2015 with melanoma, which is spread into his brain. And he was treated with anti-PD-1, and he was announced in remission, that means cancer-free that time, in 2016. So this therapy basically really made a big impact in the field of cancer treatment, and become a paradigm shift the way cancer is treated. Okay. In fact, Arlene Sharp, who is co-chair of microbiology and immunobiology at Harvard, she wrote that not all patients respond to this, but for the ones that do, it has made a huge difference to their lives. There are patients over a decade ago who had an incredibly poor prognosis, and now, a decade out, they are living normal lives. That means patients are even getting benefited for decades, not just months, not just years. So I will just ask organizers, how much time do I have?
Rahul, you can take some more time,
Huh?
Yeah, yeah. I think your story is very interesting. You can take some more time.
Sure, sure. Okay, okay. So this was the main story about the Nobel Prize winners, two scientists. And now I will also little bit talk about other revolution which is happening in cancer immunotherapy. One is the CAR T revolution. As I mentioned, some cells that these are engineered cells, these are gene therapies. Basically, gene therapy are not yet available in our country in India, but wherever a part of the world, especially in developed part of the world, in US and Europe and in Asia, only in Japan, wherever they are available, they are basically curing the patient. And these two gentlemen pioneered in it. I said in the beginning, Carl June from UPenn and Steve Rosenberg from NCI. Okay. So what is this a CAR T cell platform? So CAR T cell platform is that you have a patient in the clinic, you take their immune cells out from the blood, and these immune cells you isolate in the laboratory. So upper panel is the work that is done in the lab, and the lower panel is the work done in the clinic. So it's a very good combination of the personalized treatment. So some of you might have heard what is personalized treatment. Personalized treatments are they're tailored for the individual patient. So here, CAR T cells, they're engineered cells for specific patients. That's why every time a new patient comes, you take the immune cells out from the patients, take them to the lab, genetically modified. So genetically modification, some genetic recombinant technologies are used here. You do the recombinant technology here and then expand the cells. These are engineered cells now, which gained the ability to recognize the tumor cell. And once they gained the ability to recognize tumor cell, they reinfuse them back into the patient. And once they go back in the patient, they try to find out the tumor, act as a guided missile, and kill the tumor cell. And this treatment, unlike, as I said, the personalized treatment in patient is specific product. So one product, one patient, unlike other conventional drug, which you see in the pharmacy, where so many drugs, but one drug can be used for the different individuals. Okay. And this drug, which is CAR T cell, it was first given in 2000, I think, 12, what you see here, 2012, to a 6-year-old Emily Whitehead at University of Pennsylvania by Carl June. And this drug basically saved the life of this little kid. And now, in 2021, several years, almost a decade later, that the girl is still cancer-free, indicating this wonderful revolutionary treatment that can save the patient's life, and many patients are saved. And this drug was approved in 2017 in United States. But the cost of this drug is almost 4 crore rupees for one patient. Now, all of you who are students, whoever, you can imagine in country like us, hardly this amount, majority of the people cannot pay. Only very few can pay, right? So in US, this is like, these are in US dollar, cost is almost $500,000 US. Okay, so cost is huge, and that's the reason these drugs are not available at any price in our country right now. If person or patient need to have these drugs, they have to fly, okay, fly abroad. So this story again, I will tell you how this CAR T cells are developed, and this might motivate you, some of you at least, that in any work, whatever you do, okay, there are always tragedies, perseverance, and chance, right? So that's the story of the CAR T cells. It is published in one of the very important journal called New England Journal of Medicine. Okay. So it all started in 2005 when the first 3 patients were treated. The data was published in 2011. 2012, Carl June and Novartis is one of the pharmaceutical, they made a collaboration. And 2017, the drug was approved. This is also a very fast approval of any drug given. In 12 years, the drug was in the market. Okay. So what was the story? So the story is about this girl, Emily Whitehead, the first girl who was treated on the cancer. Okay. This girl in 2005, the 5-year-old Emily was diagnosed with BALL. Okay. That's the blood cancer. So for you all, just think it's a type of a blood cancer. This girl was treated with uh, was treated as a normal because with a chemotherapy drug, because chemotherapy works wonderfully in most of the blood cancer. So physician told Emily's parent that if you had to have a kid with cancer, ALL is the best one to have. Why? Because this cancer goes away by the 90% time. Cancer is treatable with the chemotherapy drug. 90% time patients can be saved. Chemotherapy works wonderfully in this particular blood cancer. That's the reason why parents told this is the best cancer to have. But cancer relapsed. Relapse means cancer came back. Cancer, once cancer gone, but cancer came back to this girl. Now she's again cancer patient. Unfortunately, when this cancer comes back, it's very aggressive cancer, and then nothing works. Okay, so uh, their doctor suggested bone marrow transplant. Bone marrow transplant, this is a transplant, and this transplant also does not have very good prognosis. It doesn't have very good outcome. So even you go under transplant, but it doesn't work in many cases. Okay, so and that time CAR-T trials were not there, right? So when there were no option, then oncologists are the clinicians who treat cancer. They recommended hospice or palliative care. So palliative care is you go home, whatever the symptoms are, you take those symptom medicine, and ultimately patients die. Okay, and as a father of a 5-year-old, it does not make really sense that you see your kid dying in front of you. Correct? Because and you can't do it. You're only giving symptomatic treatment. If she says I am having pain, you give pain medicine, but you are not really treating cancer. You're just waiting the kid to simply see dying. Right? So here is the Carl Jung, the clinician who treated the first patient. Right? At his home also there was a lot of tragedy. 1996, his wife was diagnosed with ovarian cancer. Okay, he tried pharmaceutical company to provide the tools to attempt immunotherapy, because in his lab he has developed this CAR T cell therapy. But whatever we develop in the lab, we cannot use directly on the patient. We have to undergo a proper regulatory trial, regulatory approval. So there are agencies which see your data and give you approval. And we also need funds to do some of these kind of clinical trial because they are very expensive. So his wife actually passed away in 2001. And then June decided to develop cancer therapy by creating a biotech infrastructure within the academy. So he then decided, I have something which he believes in, CAR-T technology. So he said, I will develop a small lab and I will try it myself. Okay. And in one interview, he said the field had been dodged by skepticism and setbacks, and the NIH wouldn't even fund a clinical trial. So many hurdles, and many times I almost had to quit. Because the reason why he was so frustrated, because many cancer treatment came, many scientists came with their hypothesis, with their new drug, and NIH or any funding agencies will give them the money to do clinical trial, but clinical trial fails in the clinic. When clinical trial fails in the clinic, it's basically if they are sponsored by the government loses money. If they're sponsored by the pharma loses money. That's why many cancer trials failed, and that's the reason why field was full with the dodged with the skepticism. Nobody believes when scientists say that I have found a new treatment. Same thing happened with him here. Then nobody will believe him. He was the only one who was like saying that yes, it works, but nobody will buy it, right? So even as he write, so many hurdles. So one of the things you all student can imagine is not just you have to keep trying, never quit. That's the message. Okay? But tragedy propels the research forward. So in one of the meeting, he met one billionaire called Barbara and Edward Netter. Now, in fact, there's a Barbara and Edward Netter Foundation. These gentlemen, they're also from the similar region, Philadelphia, and they gave them $1 million because their daughter-in-law actually died of breast cancer. So a lot of tragedies happen, and somehow they all came together, right? It's all the luck. Sometimes luck also works. And they gave them $1 million, and that was just enough to have that infrastructure, to have only 3 patients treated. And that became the history. And that led to the success, because out of 3, 2 achieved complete cure, complete remission. Remission means in cancer, cancer free, completely cancer free. He published his work, and then NCI, and now everybody funds him. Now he's a star actually. If you type the Carl June, he just, I think few weeks ago, he got $1 million prize from someone. So now he has a lot of like success, right? Novartis licensed his CAR-T technology, and now, as I mentioned, this technology is sold for one patient $500,000 US. Very expensive, but now saving many, many lives. So this gene therapy, they are not available in LMIC, low middle income countries. So low middle income countries like India, Africa, South America, those, but only available in the developed part of the world, which we all, as you all can imagine, our frustration that when the intent is to cure the cancer, right? Why there's a geographical disparity? At least some, those who can afford, they can at least get these kind of treatment, right? So cancer incidence is on the rise in our country. So many kids are having blood cancer. They can get benefit, but because this therapy is not available in our part of the world, nobody can have it, right? So majority of our patients, they choose palliative care and they don't survive. Okay. So our efforts, my lab, and we are working together with Tata Hospital in Mumbai, with the help of National Cancer Institute in US. They are our knowledge partner, and we also co-founded one pharma Immunoact for the clinical translation, with the help of all these wonderful colleagues like Dr. Gaurav Narula, Dr. Hasmukh Jain, Dr. Nirali Shah from NCI, Dr. Terry Fry, and Dr. Stephen Heifel. We come together to make this CAR-T dream, somehow bring it to India. Okay. So in summary, what we have done, we have developed a new product. We checked its efficacy, the drug efficacy. Now in the COVID era, again, many of you might have heard vaccine efficacy or vaccine toxicity. So these are the things you have to do before the going to the clinical trial. So similarly, we also have done it. We have patent, and now we are going first to the clinic. And the work is also published in one of the good journal, American Association of Cancer Research. Okay. And now this work is our work in India. Clinical trials are open. We are recruiting now the patients, like again, like the vaccines. You might have seen they are, and now they undergo clinical trials. So now some of you understanding what is called clinical trial by this COVID-19. I don't need to explain you everything. But so our trials are also open just last week. And some of our work is featured in various different platforms, including Nature Biotechnology or the AACR, this Molecular Cancer Therapeutics, which published our work, and some of the Indian newspaper also featured our work in Hindustan Times or Times of India. Okay, and thank you so much to all of you for patiently listening. And this all work is done by the wonderful colleague in the CAR-T team, Alka, Athar, Ankesh, and others. And we collaborate hugely with Sanjeeva as well as with folks from Tata Hospital, Mumbai, NCI, and other colleagues in the department. Thank you all. And if you have any questions, I will be happy to take few. Thank you.
Thank you so much, Rahul. It was a wonderful lecture. I'm sure students are, and you really made it, to the knowing the audience to really basic, which was appreciable. You have really given like a story, which many people can follow. So I think let's take the questions. Rohit Katimani, he want to ask you a question. Rohit, you can unmute yourself.
Good morning, sir. Sir, thank you for the amazing talk, sir. I have a doubt, sir. Sir, what is the efficiency rate or the success rate of this therapy, sir?
Yeah, so success rate is approximately 80% for the short term. And short term is generally in any clinical trial or in these objective response rate, which is called ORR, that is done for the 3 months. But For 5 years, data is also there where the success rate is almost 50%. So 50% of the patients for sure, they are under remission or under cure so far by this therapy for in last 5 years follow-up. So almost half of the patients can be completely treated. Yes.
So Rohit is only in class 9th. And,
Oh, okay.
and next question is from Maitri, who is in class 10th probably. So Maitri, you can unmute and ask the question.
Yeah, thank you for this lecture, sir. Sir, we say that radiation increases our risk of cancer. So in radiation therapy, that radiation will kill the cancer that we have now, but won't it increase the risk of future cancers?
I see. So it depends actually what kind of radiation is giving. Nowadays, when you take the radiation is given very localized. It is not giving whole body radiation. So whole body radiation may have chance to cure your one cancer at particular site. However, when the radiation is given localized, very precisely given at a particular area of tumor, then you have less chance to develop getting exposed at the other body parts. Only your cancer get exposed. Okay, but you are absolutely right that there is always a chance of getting exposed by any radiation. For example, even CT scan or X-rays. You might have heard now recently in this COVID era, there's a lot of people undergoing CT scan, and there's already a concern raised by Indian Medical Association and many good experts that do not expose yourself unnecessarily to the CT scan. So any radiation, not only radiation therapy, any kind of radiation is harmful to the body. But you have, again, for anything in the clinic, you have to do the benefit versus risk. If your benefit is more, for example, in cancer treatment, if you are given the radiation and radiation is helping you to kill the cancer cell, and so doctors has to estimate what is the benefit versus risk ratio.
Yeah, so now next question is from Mahima or Mahim Dwivedi, the way. Mahim, you want to unmute yourself?
Yes, sir. Sir, why didn't the chemotherapy worked in the relapse in Emily case while the first time it worked?
Yeah, that's also a good question. So the reason is that see, cancer try to evade the whatever the previously tried. Cancer wants to escape. So there are so many things happening in a cell, multiple pathways, multiple biological reactions are happening. So when you target one specific reactions to block the cancer growth, cancer try to take alternate pathways. Cancer try to reduce its dependency on that pathway, but take an alternate pathway. So the pathway which you block is no more functional because cancer has adapted a new way. So these are different ways of cancer adapt itself according to the environment. Whatever as pressure you give, that pressure cancer come out with, and then again it starts growing. So not only the chemotherapy, even cancer becomes resistant, and that is called resistance. So cancer become resistant to various chemotherapeutic drug. Cancer can even get resistant to the radiation. Cancer can get resistant to even the CAR T cell. So we are giving one CAR T cells against one particular target, but there would be some patients where there will be relapse. So as I said, only 50% patient get benefit. Other 50% may get relapse, and relapse can be because of that even the antigen which you are targeting, that antigen is no more there on the cancer cell. So cancer try to evade or whatever the pressure you give, pressure in the form of chemotherapy, radiation, cell therapy, immunotherapy, cancer come out with it and try to find alternate pathway to choose for its growth. Okay, so cancer has this beautiful mechanisms for evading any kind of pressure you give in any different form.
Next question from Kanak Sangvi. Please unmute and ask.
Thank you, sir, for the wonderful lecture. It was really nice. So my question is, can immunotherapy be combined with other treatments to make it more effective?
Yeah, so that's also a good question. So there are many modalities are tried in combination. In fact, as we all believe now in the cancer treatment, combination is the best way to do it. Go for it, because only one type of treatment when you give, that cancer try to evade it. Cancer try to find an alternate pathway and relapse. If you at the same time, if you try to combine few treatments, that will be more effective. So for example, even in the case of this PDL1, or in fact, once the discovery of this PDL1 or CTLA4 happened, now almost all whatever previously was not working, it is given in combination with PD1 or CTLA4, and they started working. So let's say if the vaccine, which were not effective in the clinic, uh, now those vaccines are given in combination with PD1 or CTLA4, they are showing very good results. Oncolytic viruses, which were alone not sufficient, when they are given with PD1 or CTLA4, they are giving very, very good results. Similarly, many chemotherapy drugs. So now people are trying to combine chemotherapy with immune therapy, with either CTLA4 or either PDL1. They are showing wonderful results, indicating yes, combination is the best way forward. Even CAR T cells are also tried with PD1 or CTLA4. Then we see less relapse. That means if you combine both treatment, not only the efficacy increases, even relapse decreases very much. So both are different. Efficacy, when we are saying that how many patients will get immediate benefit. Well, and if you combine treatment, let us say if the 50% was the efficacy, now more than 50 are achieved when the combination are tried. Similarly, relapse rate is less, because now when the combination is giving, tumor can't escape two or three multiple different pathways, right? One pathway is easy to escape, but more than one is difficult to escape. Tumor do escape in many cases, but it's difficult. The chances are less. So combination is the best way to treat cancer.
Rutuja, you are next.
Hello, sir. It was very insightful, the lecture you gave or you delivered. Sir, I wanted to ask whether we can combine gene therapy with immunotherapy for treating cancer, like, focusing on the root problem, that is from gene level.
Yeah. So Rutuja, cancer is many times. So if there is a monogenic disease that originates from one gene, it's easier to treat. Okay. So for example, a sickle cell disease, right? When you do sickle cell anemia, uh, then if the only one gene is involved, you fix that gene. However, in case of cancer, there are multiple genes can be involved, because there is dysregulation of multiple genes. So that's the reason gene therapy, if you're thinking about gene therapy as only single gene fixing, may not work in cancer, because even if you fix that particular gene, cancer can adopt another pathway. Okay. If you are thinking gene therapy and cell therapy, that in the case of CAR T cell therapy, yes, CAR T cells are working very wonderfully. That's also a different type of gene therapy, where you genetically modified the immune cells to kill the cancer cell. Okay, so gene therapy works wonderful in a monogenic disorder. Gene therapy, direct fixing certain gene in a cancer cell, may not be a very effective way of treating cancer. And cell and gene therapy, if you think about CAR T cells, yes, the XY CAR T cell is ex vivo genetic manipulation. That means you take out the immune cells from the patient, and in the laboratory you genetically modify them. So that strategy also actually working very nicely.
Great. Pranavi, you want to ask a question?
Good afternoon, sir. My question is that is it safe to get the cervical cancer vaccine, and does the vaccine lower the risk of getting the cervical cancer, sir?
Yeah. So in fact, that's the probably one of the only vaccine which produce excellent results, right? In the case of cancer, especially in our country, cervical cancer vaccine is one of the very, very wonderful vaccine, which is helping, because many of the time this cancer is happening because of certain viruses. And if you give this vaccine, you can get the body fighting against this particular virus. And if this virus is not there, then this virus induced cancer is not there. So cervical vaccine is a very good vaccine. It works and it's safe to take it. And I'm sure the many lives and many patients are getting benefit. And India, I think, did very good job in getting the people vaccinated, especially in the cervical cancer.
Thank you, sir.
All right, Manisha Thakur.
Hello, sir. Sir, my question is, does we can actually increase our immunity at any age by eating some herbal thing? And so another one is how cytokine storm can actually leads to heart attack like complication.
So first question I heard properly. Second question you may repeat, but let me answer your first question first. So do herbal thing can have the immune modulatory properties? So see, in the modern medicine, okay, evidence-based medicines are basically in very good concept for the modern medicine. You have to have evidence for the effects, and evidence for any drug is generally obtained by using the clinical trials. If any herbal medicine has gone under a proper trial where it has shown that it improves your immune modulatory properties, officially you can say yes, this herbal is good to take. Okay. However, so this was official statement, right? Because you have to generate evidence based on the clinical trial for any medicine to say that yes, it works and it definitely has a good effect. However, sometimes in our Indian science and Indian our tradition, instead of science, in our tradition, yes, uh, herbal medicine or herbal items are taken, herbal foods many times, or some spices. They have some data that these spices or these herbal products, they improve your immune properties, they have less harm. And as a prophylaxis means you can take before the disease and you may have a better immunity to fight against them. But as a therapeutic agent, I am not sure how much they will work. So prophylaxis means you can, let us say, in your healthy life and everyday routine, you can use herbal supplement. Most likely they will be good for you. But once the disease happens, let's say corona happened or COVID happens, and now you are thinking like just by taking herbal supplement, you will cure the patient. Or if patients will be, or cancer patient, you give simply herbal medicine and the patient cancer will go away. I would not recommend it. I would rather say you go and take the clinician help. That time just herbal supplement is not enough, probably because we don't have good evidence that they will work. But as a prophylaxis, everyday routine life, yes, one can take if they believe, like in Chyawanprash, for example. If somebody believes in it, please go ahead and take it. No harm in it.
All right, next question from Rashi. We'll just take maybe, Rahul, questions are not ending actually, more and more are coming in. We'll just take last two questions.
Good morning, sir. So your talk was very insightful. So my question is, can cancer be genetically prevented by manipulating genes in the laboratory? Like, is there any way we can prevent it by that?
Yeah. So yes, that's the, I also said one or two questions earlier. Cancer is now known as a genetic disease for sure, because there are a lot of mutations happen. But because there are many genes involved in a cancer development, fixing one or two gene is not enough. So one good example is there is a cancer of the skin called melanoma. In melanoma, there's one mutation in one particular gene. Okay. People have targeted that particular mutation and developed a drug. They give this drug that target one particular mutation to this cancer patient, and within 3 months, cancer was completely gone. 100% cancer gone, cancer free this patient happened. But another 6 months later, cancer came back with more aggressive form. This shows that even targeting 1 or 2 genes is not enough in case of cancer, because cancer is a disease of the, they call group of the diseases. Cancer is not one disease, but it's a group of diseases, right? So, and this not just a disease of one gene, not monogenic, it is a combination of many things. So there, only gene therapy targeting one gene would not help in the case of cancer. Cell and gene therapy, which we just discussed today, like CAR T cell, it is a good form because it completely kills the cancer cell by immune cells.
All right, one last question from Sakhi Bad, if you want to ask a question.
Yes, sir. Thank you for the wonderful lecture, sir. So I just wanted to ask, is there any cure for PVNS in the left knee with CAR T therapy?
Can you repeat? Is there any?
Sir, is there any cure for PVNS in the left knee with the CAR T therapy?
I couldn't understand what is.
She is trying to ask some disease PVNS. Can you explain what PVNS is, Sakhi?
Yes, sir. PVNS is pigmented villonodular synovitis. It's like it's a tumor only, but it's in a mass overgrowth and it keeps on recurring. So even if you get operated, it's very rare disease, but it keeps on reoccurring. And so I want to ask if it can be treated with CAR T therapy.
Yeah, so CAR T therapy, Sakhi, is not yet successful in every cancer. Right now, the data is only for certain type of blood cancer, including leukemia and lymphomas. Okay, CAR T is not being demonstrated in other solid tumor yet. There are clinical trials happening on CAR T cell with prostate cancer, glioblastoma, breast cancer, but nobody, in my actually knowledge, synovitis, nobody is doing it.
All right, Rahul, thank you so much for a wonderful lecture and giving so much valuable insight to the participants. I'm sure you can see they have so much curiosity, and probably that is the best rewarding experience of the summer camp, to see the the young minds and a lot of curious questions. So thank you so much for sparing your time. In fact, I slightly canceled 1 or 2 small session in between, but I thought to let your session continue so that they can get more benefited from these interactions. So wonderful. Thank you, Rahul. We will, if you have any other questions, student, you can put in the chat box. Maybe some of the questions I can send to Rahul and he can reply back to you.
Okay, thank you so much. Thank you, everyone. I really enjoyed a lot giving you the lecture as well as talking to you. Thanks a lot and good luck to all of you. Don't hesitate if you have any other questions, and